So I was talking about the partial agonists. So partial agonists, they have the full affinity, but intrinsic activity is between 0 to 1. While if we see the inverse agonist, what they have full affinity, but their intrinsic activity is in the negative direction. That means 0 to -1. That means they are going to produce effect just in the opposite direction to the antagonist. So the point to remember is that when we talk about antagonists do not produce any response, but it is actually the inverse agonist which is going to produce a response in the negative direction. That means it will reduce something. Like antagonist, it will not produce any effect, no response will be there. So this is very important to remember, that it is the inverse agonist which will produce effect in the opposite direction. Like beta carbolin is inverse agonist for benzodiazepine receptor. That means it is not going to allow the benzodiazepine receptor to activate, but it will further inhibit also the benzodiazepine receptors. So this is the summary what I was talking about agonist, antagonist. So when the drug comes and binds to the receptor, secondary messengers are released which binds to the effector, and they are going to produce the action. So in the agonist, the affinity is +1, intrinsic activity is +1. The receptor is basically in the activated stage. Example is the agonist. Antagonist is the one where there is affinity +1, but the intrinsic activity is 0. So both activated and inactivated state of receptor is present. Inverse agonist, affinity is +1, intrinsic activity is 0 to -1. That means in the negative direction. And partial agonist, where there is affinity is 100% +1, intrinsic activity is 0 to +1. So coming to the different receptor families, there are 4 types of receptor family. First is the ligand-gated ion channel or the ionotropic receptors. Second is the G protein coupled receptors, also known as the metabotropic receptors. Enzymatic receptors or the tyrosine kinase link receptors. And the fourth are the nuclear receptors or the receptor regulating gene expression or the steroid receptors. So what are the difference between these receptors? I will be explaining you also. The ligand-gated ion channels, they are present on the membrane. G protein is also present on the membrane. Enzymatic receptors are also present on the membrane. While the nuclear receptor, they are present intracellularly. The effector in the ligand-gated is ion channels. Ion channels or enzymes are there in the G protein also. Enzymatic effectors are there in the enzymatic one, and gene is the effector for nuclear, producing the nuclear response. And the receptor is coupled to the effector in the ligand-gated directly. That means the receptor is directly linked with that of the ion channels in the ligand-gated one. Example is the nicotinic receptor, which I have shown you, where acetylcholine was directly binding with the nicotinic receptors, and there is influx of sodium. So that type of receptors are known as ligand-gated ion channel. Then in the G protein coupled receptors, G is GTP activated protein. So it is GTP which is bound in the intracellular site, which then after the drug binds to the receptors, it produces conformational changes, activation of this G protein, and this G protein then through different pathways is going to influence the ionic channels or the enzymes. Example is the muscarinic cholinergic receptors. In the enzymatic receptors, the enzymes are coupled directly to the membrane receptors. Example is insulin. And while the nuclear receptors, the DNA in that is linked to the receptors, provides the site, and it is the gene which expresses and produces different types of protein and responses. Example are the steroid hormones. So coming to the signal transduction in the various receptors, how these receptors are going to produce the effect. So first example is the first type of receptor. It is the ion-gated receptors. They are present on the cell membrane, and they are coupled directly to the ion channels. That means the effector is the ion channel. Their onset and offset is very fastest. That means in milliseconds, because the receptor is directly linked to the effector. Ion comes and bind to the receptors, and it leads to the opening or closing of the channel based on the different drugs. If an agonist binds, the channel opens, and for example, this is a sodium channel. Sodium channel opens. There is hyperpolarization or depolarization, and cellular effect is produced. While if an antagonist is given, it will block the influx of the ion, so permeation of ion is blocked, so no cellular effect will come. So this is the example of the ion-gated receptors. Example, I have shown the nicotinic cholinergic receptors. Here, the two molecules of acetylcholine binds and produces conformational changes in these receptors, leading to the influx of sodium and efflux of potassium. So this is the example, that is nicotinic cholinergic receptors is a type of ion-gated chain receptors. Next is the G protein coupled receptors. These receptors are membrane bound and they are coupled to the G protein, which is GTP activated protein. What happens? These receptors are present in the cell membrane. The agonist comes or the antagonist comes and bind to these receptors, and on the inner side of this membrane are present the G protein. The binding of agonist will lead to the and this G protein is the secondary messenger over here. It gets activated, then it goes and binds to the effector produces effector protein and then produces the effect. This is how G protein coupled receptors act. This is the structure of a G protein coupled receptor, which is having the 7 helical structure. Then amino terminal is external and the carboxy terminal is internal and intracellular, and agonists bind in between over here. So the different types of G protein are G stimulatory protein, G inhibitory protein, GQ and G0. G stimulatory acts by increasing the adenyl cyclase activity, thereby increasing the cyclic AMP level, and also opens the calcium channels. Examples are the beta adrenergic or glucagon or S2 receptors. G inhibitory receptors is the signal pathway or the effector is just opposite to that of the G stimulatory, where they inhibit the adenyl cyclase and thereby decreasing the cyclic AMP or open the potassium channel. Example are the alpha 2 adrenergic, M2 type of muscarinic receptor. Then GQ receptor acts to the phospholipase C inositol triphosphate diacylglycerol pathway. These I am going to show you in the diagram also how these drugs are going to act. Example is the alpha 1 receptor, G0 is M2 or the D2, which acts by inhibiting the calcium channels. So the basically G protein coupled receptors, they follow three pathways. One is the adenyl cyclase pathway through which the G stimulatory and the G inhibitory protein acts. G stimulatory activates this pathway, G inhibitory they inhibit this pathway. Then GQ acts through the phospholipase C inositol triphosphate diacylglycerol pathway. And channel regulation occurs with all these G stimulatory, G inhibitory, GQ as well as the G0 G proteins. So in this diagram, it appears to be a bit complex diagram, but it is actually the cyclic AMP system, where if you see this beta receptor, which is the adrenergic receptors, when adrenaline comes and bind to this receptor is a GS coupled receptor, that is G stimulatory. What happens when the agonist comes and bind to this receptor, then there is hydrolysis of GTP, and on the inner surface of this receptors, it is bind to the G stimulatory G protein, type of G protein. It has got three subunits, alpha, beta, and gamma. The hydrolysis of GTP after binding of the agonist, what it does, the G stimulatory alpha moves and binds to the adenyl cyclase enzyme, which is present on the inner surface of the biological membrane. This leads to the conversion of ATP into cyclic AMP, and this cyclic AMP is the secondary messenger which leads this whole diagram is that of a cardiac cardiac myocyte. So cardiac muscle. So this will ultimately leads to increased cardiac contractility and relaxation, thereby leading to quick contractility and relaxation. What these GS alpha unit also do, they will open the calcium channel. Cyclic AMP also opens the calcium channel, and even the protein kinase, they also open the calcium channel. There is increase in the calcium, which will lead to increased contractility, and this is how the G stimulatory they are going to act through the cyclic AMP pathway. Then coming to the G inhibitory on the myocardial cells, the cholinergic receptor, it is the cholinergic receptor, M2 type receptor, it is going to inhibit the cardiac myocyte. So what it does, it acts to the G inhibitory one, where there is removal of the beta and gamma subunit by the hydrolysis of GTP, and this activated G inhibitory then go and bind with the adenyl cyclase, and they inhibit the activity of the adenyl cyclase. The cyclic MP level decreases. So these all chain reaction decreases, and there is inhibition of the contraction of the muscle. So that is how the G inhibitory protein then acts. Then next to this is the second pathway, that is the phospholipase C pathway, through which the H1 histaminergic receptors act. What they do, after the binding of histamine to the histaminergic receptor, and this is a bronchial smooth muscle. What histamine has the effect on the bronchial smooth muscle is it produces the contractility of the bronchial constriction, of the bronchoconstriction it causes. So what happens after binding of the histamine to its receptors, GTP gets hydrolyzed and GQ protein is activated. It goes and binds to the phospholipase C, which is present on the inner side. This leads to the hydrolysis of phosphoinositol diphosphate into two molecules. One is inositol triphosphate and other is the diacylglycerol. Inositol triphosphate is water soluble, it moves in intracellularly, but diacylglycerol it remains in the membrane and it activates the protein kinase C, which leads to the further protein phosphorylation. Inositol triphosphate then increases the calcium release from the sarcoplasmic reticulum, and this increased calcium binds with the calmodulin and the myosin light chain kinase. What it does, it produces the contraction. So that is how this bronchoconstriction occurs with the histamine, that is GQ coupled protein. Third, regulation of the G protein is regulating the ion channels. So I have told that GS opens the calcium channel in the myocardium and skeletal muscle, and G inhibitory or G0 opens the potassium channels in the heart and smooth muscle, as well as inhibits the neuronal calcium channel. So this is how the G protein coupled receptors can control the functioning of the ion channel by not involving any secondary messenger. So these are the different types of G protein coupled receptors when you will be taught about the respective topics. So then you will be more aware of how these receptors they are going to act. Coming to the next type of receptor family, that is the enzymatic receptor. So how enzymatic receptors? These receptors are directly linked with tyrosine kinase, which is present intracellularly, and insulin receptors are the enzymatic receptors. These receptor binding domain is present extracellularly, and they produce the conformational changes in the intracellular side. So how these receptors act? That after the binding on the receptor side by these insulin, these receptors they undergo dimerization. That means two receptors will come close, and there is a catalytic side intracellularly which is having tyrosine kinase activity. By dimerization, what happens? They undergo phosphorylation, and then they are going to further phosphorylate certain sulfhydryl proteins, and these phosphorylated proteins are then going to produce the intracellular changes or production of different types of protein or signals. So this is how the enzymatic receptors they are going to act. Another type of enzymatic receptor is the JAK-STAT binding receptors. and they are different from the previous receptors, that is from the insulin receptors, because their intracellular side they have the tyrosine, but these are not having any after dimerization they do not undergo phosphorylation. So JAK stands for Janus kinase. When these through these receptors, cytokines, growth hormone, prolactin, and interferon acts. So what they do after the binding of the cytokines, they go dimerization, and from the cytoplasm, then Janus kinase comes and bind to the uh, their tyrosine residues, and then stat from the cytoplasm they come and bind, and they have the tyrosine residues which undergoes the phosphorylation, and then this is stat dimer then separates out from the receptors, and it produces the effect by transcription of the genes or produces the signal molecule. And so this is somewhat different from the previous enzymatic, that is the insulin receptors. Last type of receptor is the nuclear receptors, where due to the binding of this heat shock protein 90, this is having a different conformational structure. When glucocorticoids come inside the cell or the steroid, they bind to the carboxy terminal, they produce the conformational changes, and these dimers the receptors undergo dimerization and they move to the nucleus. They bind to the gene response element of the DNA, and they are going to produce certain response. They undergo transcription, producing the mRNA, and the mRNA then produce the different types of protein, and they modify the cellular function. So this is how the gene expression is regulated. So the rest, this was the all about the different types of receptors. Then receptors can undergo their own regulation by if they are having a dynamic state. So when there is prolonged use of any agonist, the receptors they decrease in number and sensitivity, thereby decreasing the drug effect. So chronic use of example is chronic use of salbutamol, which acts on the bronchial smooth muscle and used in asthma. They downregulate the beta 2 adrenergic receptors. So thereby their bronchodilatory effect goes out by chronic use. Similarly, prolonged use of any antagonist will lead to the increased number of the receptor and sensitivity, and the drug effect will increase. Like if a person is taking propranolol for a longer duration, so when propranolol is stopped, there will be withdrawal symptoms. So propranolol decreases the blood pressure. So what will happen? The propranolol is going withdrawal will lead to very rapid shoot in or increase in blood pressure or induce angina. So that was all about the receptors. Thank you very much for your patience.